Okay, let's get started. Hello everyone, good morning. And now we start muscle tissues. As you remember, we have four types of tissues: epithelial tissues, tissues of internal environment, muscle tissues, and nervous tissues. In this block, we will discuss muscle and nervous tissues, only two topics in the third block. And you will have the colloquium, which consists only of tests, no oral part about these two types of tissues, but still you need to know everything about these tissues to have the signatures and to pass the topics. So, muscle tissues include striated muscle tissues and smooth muscle tissues. In turn, striated are subdivided into skeletal and cardiac muscle tissues, and smooth muscles include just visceral smooth and myoneural smooth muscles. By structure, muscle tissues are divided into striated. Striated means um, the structures of striated muscle tissues have cross striations, the cross lines in it, and smooth muscles, which have no striations, so it just looks smooth. By location, these types of muscle tissues are subdivided. Striated one is divided into skeletal and cardiac, and smooth into visceral and vascular. Visceral means inside organs in our body, such as urinary bladder, intestines, etc. Vascular means in the structure of blood vessels, for example, in arteries. There is also smooth muscle tissue. And by mode of contraction, these muscle tissues are striated skeletal, voluntary or conscious regulating.
Э,
Yes?
Извините, профессор, не слушаю.
Aha, you do not
Не
see the
смотрит.
presentation, yes?
Yes, yes.
Okay. What about now?
Да, yes.
Okay.
Очень.
Thank you. Thank you.
Спасибо.
So, we discussed this slide, the classification of muscle tissue types by structure. We have striated and smooth. Striated have cross striations, smooth do not have cross striation. By location, striated is subdivided into skeletal and cardiac. Skeletal includes skeletal muscles, muscles of our body, of arms and legs and our body, and cardiac means in heart. Smooth muscle subdivides into visceral in organs and vascular in blood vessels. By mode of contraction, striated skeletal muscle is voluntary. It means it is regulated by our conscience. It means that we can contract our muscles of our arm when we want it. But all other types, cardiac, visceral, and vascular, are contracting involuntary. We cannot regulate their contraction if we want to. Sources of muscle tissue development include for muscles of limbs, trunk, tongue, etc. All these voluntary contracting muscles, they are called somatic, and they are derived from myotomes of somites. Somite is all this area. It includes dermatome, myotome, and sclerotome. We are talking now about myotome. Myotome gives rise to skeletal muscle tissue. The cardiac muscle tissue is called coelomic by its source of development because it develops from the coelomic epithelium. Coelom is this cavity of the embryo. Coelomic epithelium, the epithelium which lines this cavity. This area of coelomic epithelium in the frontal part of embryo is called myoepicardial plate. This name comes from myocardium and epicardium of the future heart that will develop from this area. So this is the part of the visceral layer of splangnatome. this part, and it will give myocardium of the heart, the striated cardiac muscle tissue in heart. Most of the smooth muscle, such as visceral inside the organs, here intestine is shown. It has 2 layers of smooth muscle tissue in its structure, which helps peristalsis, the propulsion of food inside intestine. And also the smooth muscles of blood vessels and of endocardium in the heart. This smooth muscle develops from mesenchyme of mesodermal origin. And also we have specific neural sources of development for smooth muscles. They include neural tube, which gives rise to muscles of iris. Iris is this part of the eye, which gives specific color to our eyes. And the function of the muscle of iris is to regulate the diameter of pupil, the dark hole in the center of our eye. And the purpose of its regulation to increase or decrease amount of light which enters our eye. And second source of neural source of development include the mesenchyme of neural crest. Neural crest is this area between neural tube and ectoderm. So neural crest gives rise to ciliary muscle. It is also inside our eye. It is here and here in ciliary body, and the function of this ciliary muscle to regulate the shape of lens. This part of the eye is the lens, and its shape changes when we want to see the objects which are very close to our eye, or which are very at long distance from our eye. In order to see such objects, we should regulate the shape of our lens with the help of ciliary muscle. And muscle tissue types, according to how they look like in histological sections, they are divided into skeletal, cardiac, and smooth muscles. Skeletal muscle, its functional and structural unit is muscle fiber, very long structure. In this picture you can see 3 muscle fibers in longitudinally section, and the same in cross section. These structures are muscle fibers. Cardiac muscle tissue consists of cells. Here you can see one such cell. It is called cardiomyocyte. These cells are combined together with the help of cell junctions, and this is how it looks like in cross striation. Also you can see here cross sections of cardiomyocytes. As you can see here, both skeletal muscle and cardiac muscle tissue are cross striated. These lines are cross striations, which give these 2 types the name striated muscle. And smooth muscle tissue has no such striations. It looks smooth, and it consists of cells which are called smooth muscle cells or smooth myocytes. Here you can see them in longitudinal section, and here on cross sections. Now we will talk about each type in detail. So we start with skeletal muscle tissue. Its structural and functional unit is the muscle fiber. Here is the muscle fiber. It consists of 2 elements, myosymplast and myosatellite cells. Most of the muscle fiber consists of myosymplast. Here you can see myosymplast, and these are myosatellite cells. Myosatellite cells are cambial or stem cells which can fuse with the myosymplast during the growth and regeneration of muscle fiber. It means that if you go to gym, for example, and your muscles start to grow, it should increase its intracellular structures, increase myofibrils inside it, and it means that muscle fiber should increase amount of proteins inside it. To synthesize proteins, you need DNA, and from this DNA then RNA is transcribed, and then from RNA you will have the amino acids combined together to form proteins. So if you need more protein, you will need more DNA. And in this case, myosatellite cell nuclei, which will fuse with the myosymplast, will increase the amount of DNA needed to produce proteins to build myofibrils. Myofibrils are specialized contractile elements or organelles inside the myosymplast. So here you can see myofibrils. They are very long. They consist of contractile myofilaments, actin and myosin, and each myofibril goes from one end of muscle fiber, from one end of muscle till another end. So they are very long structures. They are contractile elements. The inner space of muscle fiber is the cytoplasm, which is called here sarcoplasm. Sarco stands for muscle. It is Greek term for muscle. So cytoplasm is sarcoplasm. Cytolemma is called sarcolemma, and together sarcolemma with the basement membrane, it will be the membrane of the muscle fiber, sarcolemma. It includes the plasma membrane of muscle fiber and basement membrane, which covers both myosymplast and myosatellite cells. Myosymplast, as you remember, is the result of fusion of many cells, myoblasts, during development of muscle fiber. So in this picture you can see sarcolemma, outer covering of muscle fiber, and two components, myosymplast and nuclei of myosatellite cells. During embryonic development of skeletal muscle, the myotome cells become the cells which can move, myoblasts, and they start to move, to migrate. They undergo divisions, proliferation, and they go to the place where they will form the muscles. So they start to migrate in some areas of the embryo, and then they start to fuse together to form myotubules and then myosymplast. During this process, they should be coupled with capillaries and nerves, which give blood supply and innervation to form the skeletal muscle fibers. Here you can see how this happens. At first stage, you have cells, migrating cells, myoblasts, that proliferate. It means they divide via mitosis. Then, in second stage, they start to fuse together, fusion, and form myotubes, elongated multinucleated structures. Some of myoblasts do not fuse to form myotubes, but stay close to this myotube, and they will form myosatellite cells, the cambial cells. Next, myotubes grow, increase in diameter because of starting of formation of myofibrils and other organelles needed to form the inner structure of future muscle fiber. And in the end, you have muscle fiber, which consists of myosymplast, inside which nuclei move to the periphery and they stay at the periphery of myosymplast, and all the rest inner space of myosymplast is filled with myofibrils, a lot of myofibrils, with all other organelles between these myofibrils. Myosatellite cells stay very close to the myosymplast to form muscle fiber. So this is how muscle fibers look like in histological sections. Here you can see them, and at longitudinal section you can see cross striations, these lines. You can see these lines which go along the muscle fibers. These lines which go longitudinally, these are actually myofibrils. Myofibrils are the structures that are stained with eosin. So this is the hematoxylin and eosin staining, and in this staining muscle fibers look red because myofibrils are stained with eosin. Myofibrils consist of actin and myosin myofilaments. Nuclei are located at the periphery of muscle fiber, not in the center. At the periphery and between muscle fibers you see layers of loose connective tissue, and inside this loose connective tissue we will have blood capillaries and nerves. Here you can see collagen fibers and cell nuclei of loose connective tissue. The loose connective tissue between muscle fibers, very thin layers of loose connective tissue, will be called endomesoum, and as you can see, the muscle fibers are combined together to form bundles of muscle fibers, and between those bundles we will have larger, thicker layers of connective tissue, which will be called perimesoum. Here you can see the bundle of muscle fibers which are cross-sected. So this is one cross-sected muscle fiber. The stained part is the myofibrils. They are stained. Nuclei are located at the periphery. Here the same muscle fiber. Nuclei are located at the periphery. Between muscle fibers, loose connective tissue with its nuclei. So this is the nucleus of some cell of loose connective tissue, not of muscle fiber. The same in different staining, iron hematoxylin. Here you can see muscle fibers at longitudinal section with cross striations and cell nuclei at the periphery. Between muscle fibers, loose connective tissue, endomesoum. Here muscle fibers at cross section. Also nuclei at the periphery. So muscle consists of muscle fibers. Inside each muscle fiber you have myofibrils. Here is one myofibril. It is the element of the contractile apparatus or contractile organelle inside muscle fiber, and each myofibril consists of very highly organized thicker myosin, pink in this picture, and thin actin myofilaments, yellow in this picture, which are very highly organized. And the part of myofibril from one so-called Z line till another Z line. Here you can see that line, and here also Z line. This part will be called a sarcomere. A sarcomere is the subunit of myofibril. Myofibril consists of many sarcomeres. Each of them consists of everything between two Z lines. Z lines are the areas into which the thin actin filaments are attached. So the Z line is the middle line which attaches the thin actin myofilaments. So muscle fiber, you can see here muscle fibers. Inside them we have myofibrils. In cross sections they can look like very small red dots. You can see them, myofibrils inside muscle fiber. one sarcomere is the part of myofibril with thin actin and thick myosin filaments, and the area which consists of thick myosin filaments and also actin filaments, this area is called A band. It looks darker. The area which consists only of actin filaments, this area will be called I band. It looks more lightly stained. The Z line is in the middle of the I disc. In the middle of A disc we have this so-called M line. It is this area. M line is the middle part which connects thick myosin filaments in this area. If you look at A band, you see that it consists partly of the area where you have both thick myosin and thin actin myofilaments, and in the very middle area you have the area with only thick myosin filaments, and this area inside which you have only thick myosin filaments is called H zone. So at high magnification, one cross-sected muscle fiber. Inside it you can see these red dots. They are cross-sected myofibrils. This is the nucleus of the myosyplast. So a sarcomere is the part of myofibril from one Z line till another Z line. Here is also the sarcomere. In this picture, actin myofilaments thin are blue and thick myosin filaments are red. During contraction, the actin myofilaments will move deeper between the myosin filaments because the myosin filaments have these heads which attach to actin myofilaments and pull them to the middle zone, towards the M line. So during contraction, every actin thin myofilament is pulled towards the M line, like this, and these to M line like this, and this will cause the contraction of the sarcomere and the contraction of the myofibril. So myofibril is the structural and functional unit of the contractile apparatus of muscle fiber, and a sarcomere is the structural and functional unit of myofibril. Z line is the middle line of the light I band. M line is the middle of dark A band. In this area you have only actin myofilaments, as you can see here. In this area, which corresponds to this one, you have both thick myosin and thin actin myofilaments. And in the M line, in this area, in the H zone, in this area, you have only thick myosin filaments. Now we will look closer to the structure of the myofilaments. This one in this slide is thin actin myofilament. It consists not only of the protein actin, but also of some other proteins. Let's look. So actin protein is the round-shaped protein. In this form it is called G-actin. G stands for globular. Globular means round, like a ball. These balls, actin proteins, are combined together to form F-actin. F stands for fibril. So a fibril is some elongated structure. So these G-actins form the chains of F-actin, which consists of two chains, one and second one. They form this chain. Also, we have here the tropomyosin molecule, also two molecules. Here you can see one of them, and here second one, tropomyosin molecules, and the complex of troponin molecules. This complex includes three molecules, troponin T. T stands for tropomyosin because this is the part which binds to tropomyosin. Troponin C. C stands for calcium because this protein binds calcium ions. And troponin I. I stands for inhibition because it inhibits actin-myosin interaction. Here the same. So in the resting state, the troponin tropomyosin complex masks or closes the active center of the actin molecule, which is designed to attach to myosin myofilament. So in resting state, the troponin and tropomyosin molecules prevent attaching of actin myofilament to the myosin myofilament and prevents contraction. Now, thick myofilaments or myosin myofilaments. You can see here one of them. This is the middle area which attaches to M line inside the sarcomere. So here should be the M line. One thick myosin myofilament consists of very high amount of the molecules of myosin. You can see these molecules. Each molecule consists of the tail and two heads, and also with neck between the heads and the tail. Here you can see these tails. They are long structures. They start in the middle part and go to the periphery, and the heads are located at the periphery on both sides of these molecules. The heads, every of these two heads has two sites for binding some substances. This is the actin binding site. This area is designed to attach to actin thin myofilament. And this red in this picture is the ATP binding site to bind ATP molecule. ATP is needed to perform contraction. It is energy dependent process. Here you can see how these actin and myosin filaments are arranged inside sarcomere. So sarcomere is the area of the myofibril between two Z lines or Z discs. Here one and here another one. So this is one sarcomere. The thick myofilaments are attached together in the M line here. So in this area, this is how the cross-section would look like. It will consist only of thick myofilaments here. In this area, we will have both thin and thick myofilaments, thick and thin myofilaments, and in this area, only thin myofilaments. The thin myofilaments are attached to the Z line with some proteins such as nebulin, and the myosin filaments are attached to M line with the M and C proteins and also the large protein titin. Here you can see these proteins needed for attachment of thin and thick myofilaments. So this is the M line which attaches thick myofilaments together. It includes C proteins here and M proteins or myomesin proteins. These are attachment proteins to attach the thick myofilaments to M line and to bind them together. And also, as you can see here, myofilaments are attached to Z line with the help of very large protein titin. Titin stands for titan, some very giant person, because it's very giant, very large in size protein. It has this spiral-like part which prevents overstretching of muscle fiber, overstretching of fibril. So titin binds myofilament to Z line. Now, actin myofilaments are attached to Z line with the help of alpha-actinin here and nebulin molecule. Nebulin molecule, you can see it here. Also, all myofibrils, here you can see them, they are attached together along Z line. Here is the Z line. It is in the middle of the light eye disc here. So they are attached together along this area with the help of intermediate filaments, which are composed of the protein desmin in muscle tissues. As you remember, intermediate filaments consists of tissue-dependent proteins. They are different in different types of tissues. In epithelial tissues, intermediate filaments consist of keratin protein, and in muscle tissue, intermediate filaments consist of desmin protein. So, molecules of desmin combine the myofibrils along Z line, and also these intermediate protein attach the myofibrils to the sarcolemma of the muscle fiber in the area which is called castomere. Here you can see them, castomere. It is the area of attaching of myofibrils to the membrane of the muscle fiber, costamere. So the cross-striation, which is seen in light microscopy, is the result of alternating light and dark cross bands, which are the result of very highly organized location of myofibrils inside muscle fiber. Every light eye band is attached together in adjacent myofibrils, and dark bands also are attached together in adjacent myofibrils. One A band below second one and below third one. Now, the mechanism of contraction is called the mechanism of sliding filaments. So it starts with this part. Step 1 is when the actin and head of myosin filaments are not attached together. This is the head of myosin molecule. It is its neck and it is its tail. It cannot attach to actin because actin binding site is closed. It is covered with the troponin molecule. So, in order to have the ability for our myosin to attach to actin, we should remove the tropomyosin molecule from the actin. So, what happens? We need calcium ions. Calcium ions will bind to troponin C. It changes the configuration of troponin subunits, and the tropomyosin molecule releases the actin binding site which binds to myosin, and this allows the head of myosin to attach to actin. Now we need ATP molecule. The ATP binds to the ATP binding site in the head of the myosin, and now we have the bridge or cross bridge between the actin and myosin molecule. This is this cross bridge. result of attachment of myosin to actin. Now, the hydrolysis of ATP happens. It means that ATP breaks down into ADP and phosphorus. It gives energy, and this energy goes to change the shape of the myosin molecule. So it is its resting state, and this is how it looks like as the result of using of energy. Its head bends towards left side of this picture, and during this movement of the head towards left side, because our head is attached to the actin, so myosin molecule pulls all this actin chain also towards left, and this causes movement of actin filament along myosin filament. Then we will need another molecule of ATP. Another molecule of ATP will be attached to the head of myosin, which allows myosin head to detach from actin and to return in the resting state like this. And then this cycle will be performed once again. Regulation of contraction. So in order to perform the contraction, we need calcium. Calcium ions are needed, as you know, to release the actin sites on the actin molecule, the sites of binding to the head of myosin filament. So we need calcium. Calcium is stored inside the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is called here sarcoplasmic reticulum because sarco stands for muscle here. It has its specific shape. The cisterns make this network of the sarcoplasmic reticulum, which covers from outside every myofibril. So this is one myofibril, this is partly second one myofibril. They are covered from all the sides with the network of the sarcoplasmic reticulum. The same you can see here. This is this network of sarcoplasmic reticulum which covers all the myofibril. Next, we have second element, the so-called T-tubules. T-tubules, as you can see here, are the invaginations, very deep invaginations of the membrane of the muscle fiber, which is called sarcolemma. So sarcolemma invaginates and forms these long and thin tubules, which also go around every myofibril inside muscle fiber. So these are T-tubules inside muscle fibers. The same you can see here. This is the sarcolemma, outer covering of muscle fiber. It makes these invaginations, which go deep into muscle fiber and surround every muscle myofibril. And as you can see here, in this area, the T-tubule goes very closely to the parts of the smooth endoplasmic reticulum, which forms so-called terminal cisternae, terminal or lateral cisternae, enlarged parts of sarcoplasmic reticulum, which lies at both sides of the T-tubule. So as a result, we have this structure, which is called a triad. Triad because it consists of three elements. One T-tubule in the middle and two terminal cisternae on both sides of it. Another one is here, and the same you can see here. T-tubule here, and on both sides, very close to it, the L-cisternae, or terminal cisternae of sarcoplasmic reticulum. This is how it looks like in electron microscopy picture. This is the T-tubule, This is it. It consists of the membrane and basement membrane, two elements of which sarcolemma consists. And these are L cisternae or terminal cisternae of smooth endoplasmic reticulum at both sides of T tubule. So the function of sarcoplasmic reticulum, one of its functions is to store calcium ions needed for contraction. T tubules invaginations of sarcolemma need to provide electric impulse towards L cisternae to cause release of calcium ions from it in order to contract. This is how it looks like in electron microscopy picture. So you can see here the part of the muscle fiber in all this picture. In it you can see a part of the myofibril. One of them is here, second is here, third one is this one, and part of the fourth. Between the myofibrils you can see some organelles, also here organelles and here organelles inside muscle fiber. Most of these organelles are the vesicles and tubules of different shape. This is smooth endoplasmic reticulum or sarcoplasmic reticulum. This is it. These are granules of glycogen, and in this area, the Z line, in the area of Z line you can see here the triad, and also here, it consists of T tubule in the middle and two terminal cisternae of SER, the same as here, T tubule and sarcoplasmic reticulum. In this picture also you can see a part of the muscle fiber. It has a lot of myofibrils. They go like this in this direction. Between them you can see some organelles. These are, for example, mitochondria. They lie between myofibrils, and also other organelles also lie between myofibrils. In every myofibril, a disc of one myofibril is below the a disc of another myofibril and another myofibril, and the same for light I discs. So we have this cross striation. We have darker lines and lighter lines as a result. And now we look to a triad. It is shown here. So it consists of the central number 2 here, T tubule, and L cisternae or terminal cisternae of smooth endoplasmic reticulum, number 3 in this picture. They together make a triad. It is the feature of the skeletal muscle tissue. And now how all this works. So in order to contract muscle fiber you need a nerve impulse from a nerve cell, because the contraction of skeletal muscle is voluntary. It means it is regulated with our conscious, with our brain. If we want to contract our muscles in our arm or leg, for example, it means that we will have here the contact between the end of the process of a neuron cell, this is the axon of a neuron, and muscle fiber. So this is the area of their attachment, of their contact, which is called motor plate. So inside the terminal of the axon we have the synaptic vesicles with neurotransmitters. Acetylcholine. ACH stands for acetylcholine. This is the neurotransmitter which needs to contract skeletal muscle fiber. So step 1, neurotransmitters come out from the terminal part of the axon, and they bind to their receptors, which are incorporated in the membrane of the sarcolemma. So these are receptors. Acetylcholine binds to these receptors. Step 2. Binding of acetylcholine to receptors cause depolarization of the membrane of the muscle fiber. Depolarization means change of its charge. In resting state, we have more positive charge outside, so here will be a lot of plus, and inside we have a lot of minuses, so negative charge inside the cell. Change of charge means that after change we will have pluses inside, minuses outside. So depolarization means change of charge of the membrane. And step 3, this change of membrane starts to spread along the membrane, including its invaginations, which are called T-tubules. T-tubules are the invaginations of the membrane. So this wave of depolarization goes along T-tubules inside muscle fiber. Next, step 4. In this area, as you can see here, the L cisterns, terminal cisterns of sarcoplasmic reticulum, come close to T-tubules, L cisterns inside which calcium are located. So in L cistern membrane we have voltage receptors that sense the change of the charge of membrane of T-tubules. And when these receptors feel that the membrane charge of the T-tubule changed, it will cause step 5, the open of the calcium channels inside the membrane of the smooth endoplasmic reticulum. And step 6, these channels open and calcium can go out from smooth endoplasmic reticulum. So calcium ions go out from smooth endoplasmic reticulum to the cytoplasma, which is called sarcoplasma here, and calcium goes towards myofibrils. There calcium will bind to troponin C and it will launch the contraction of myofibril. So this is step 7, and when the contraction is finished and we want to return to resting state, we need all the calcium to go back to its storage place, to the smooth endoplasmic reticulum, and this happens with the help of the calcium pumps, ion pumps located also in the membrane of smooth endoplasmic reticulum, which pumps calcium inside the smooth endoplasmic reticulum from sarcoplasma. And during next contraction, all will be the same. It will start with the release of neurotransmitters from neuron cell, which will act on the receptors upon the surface of the muscle fiber. So in this picture you can see the inner structure of muscle fiber with all the labels. Now we talk about the structure of skeletal muscle as the organ. So we have our skeletal muscles, biceps for example in our arms. As all organs, it consists of several tissues. The major tissue here will be the skeletal muscle tissue. It is organized in the muscle fibers. So here you can see muscle fibers. Here they are. Every muscle fiber is surrounded with a very thin layer of loose connective tissue with blood capillaries and nerves, which are needed for life of muscle fiber and for its contraction. And this thin layer of connective tissue is called endomysium. Next, our muscle fibers are combined to make the bundle of muscle fibers. Here one bundle, here second, here third one. These bundles are separated from each other with a more thick layer of connective tissue, which is called perimysium. It surrounds bundles of muscle fibers. And muscle as an organ is covered from outside with thick layer of connective tissue, which is called epimysium. It will contain dense fibrous irregular connective tissue, outer layer, and inner layer loose fibrous connective tissue. So epimysium, perimysium, and endomysium resemble the epitendinium, peritendinium, and endotendinium in the tendon. As you remember, tendon, there will have bundles of collagen fibers of different orders, also separated from each other with layers of connective tissue. Here the same, but here we have not bundles of collagen fibers, but the muscle fibers. We have three types of muscle fibers according to their biochemical and physiological properties. First class is called red muscle fibers. They are slow, fatigue resistant. It means that they can contract very long periods of time without being tired. So they can not be tired very long time. They are oxidative. It means that they use a lot of oxygen to contract, and they contain a lot of mitochondria. Mitochondria use this oxygen. And also they contain a lot of myoglobin. Myoglobin is the oxygen-binding protein which resembles hemoglobin inside erythrocytes, which is also oxygen-binding protein. So these red muscle fibers prevail in some muscles that should contract for a very long period of time without being tired. For example, those which maintain our posture, our pose, when we stand or sit in the same pose. For example, long muscles of our back. And also these muscle fibers are found in other muscles in our body, and they prevail in, for example, in marathon runners' muscles. So in persons who can run for a very long period of time and not being tired. In this picture, red muscles fibers are these deeply blue stained. This is specific staining for the proteins of the oxidative chain in mitochondria. So these will be red muscle fibers. White muscle fibers are fast. They can contract very fast, but they are fatigue prone. It means that they get tired very fast. They are glycolytic, so they perform glycolysis. They contain a lot of glycogen and few mitochondria, and they prevail in some muscles that should contract very fast to ensure very thin and precise movements, such as muscles that move our fingers, and also extraocular muscles that allow our eyeballs to move when we look at some paintings, for example, and when we read. These muscle fibers are here, white. And also we have intermediate muscle fibers. They are something in between those two first. In this picture it will be here. Now, as I said, every muscle fiber, here in this picture we have 3 muscle fibers. Each of them needs its blood supply with blood capillaries, individual blood supply, and individual innervation. This is the end part of the nerve, which goes to each of these 3 muscle fibers. And together one muscle fiber with its blood capillary and its motor plate, the end of a nerve cell, will be called a myon. It's the structural and functional unit. So here you can see the scanning electron microscopy picture of several muscle fibers innervated with the somatic nervous system. These are the nerve fibers. And here another functional unit, which is called motor unit. Motor unit is a motor neuron with all muscle fibers that it innervates. Motor neuron is a nerve cell. Here is its body. It has small dendrites and very long axon. Axon can branch in its end portion, and every branch will innervate some muscle fiber. So one motor neuron, which cell body is located inside the spinal cord, and all the muscle fibers that branches of its axon innervate. So all of this will be called motor unit. These are motor plates. Motor plates, the site of contact between the end of axon and muscle fiber. So here it is. It's the area from which acetylcholine exits and acts upon receptors of upon the membrane of the muscle fiber to launch the contraction. So we see striated skeletal muscle tissue in the section of esophagus. In esophagus, here is its lumen, lined with stratified squamous non-keratinized epithelium. Here is it. It has loose connective tissue under the epithelium. Also, you see here the glands, which are mucous glands. They produce mucus, which lubricates the epithelium to ensure the passage of food through esophagus. And finally, we have here muscle tissue, which lies here in two layers, inner layer and outer layer. This is at low magnification. So here is the epithelium, connective tissue, glandular epithelium, mucous glands, and here muscle tissue, two layers, inner layer and outer layer. At high magnification, this is what we see. This can be inner layer, this can be outer layer. Both consist of bundles of muscle fibers. Each muscle fiber is separated from another with thin layer of endomysium, bundles of muscle fibers. Here one bundle, here second bundle, between them thicker layer of connective tissue, perimysium. Also, we see striated skeletal muscle tissue in the section of the tongue. Most volume of our tongue actually consists of skeletal muscle tissue, inside which we see the bundles of skeletal muscle fibers which go in different directions. Basically, they are organized in all three directions, and this allows our tongue to move in any direction we want. So, in this section stained with iron hematoxylin, we see in some areas cross sections of muscle fibers, in some areas longitudinal sections of muscle fibers, and in some areas oblique sections. Longitudinal means along some long structure, cross section means across, oblique means like this. So here longitudinal sections of muscle fibers. These are muscle fibers because you cannot see here the borders between the cells, because muscle fiber is not a cell, it is a multinucleated structure, and cell nuclei are located at its periphery. This can be the nucleus of myosymplast, and this can be the nucleus of myosatellite cell. In this area you can see cross sections of muscle fibers, one here, one here, one here. Also, nuclei are located at their periphery, between muscle fibers, endomysium, between bundles of muscle fibers, perimysium. Now we go to second type of striated muscle tissue, cardiac muscle tissue. It is located inside the heart, so this is why it is called cardiac. Basically, it is located in the heart's thickest layer, which is called myocardium. Myo stands for muscles. So the cavity inside the heart, you can see here its ventricle cavity is lined with the thin layer, which is called endocardium. In the histological section, it is very thin line here, lighter stained. Then goes very thick myocardium, which consists of the cardiac muscle tissue. It is the thickest layer of the heart's wall. You can see it here. Everything which is eosinophilic. In this picture, histological section, this is myocardium. And the outer layer of the heart's wall is called epicardium. Epicardium consists of loose fibrous connective tissue and adipose tissue. It is this area. The function of cardiac muscle tissue is to contract and to pump the blood out from the heart to aorta and the pulmonary arteries. Structural unit of this muscle tissue is the cardiomyocyte. It is a cell, so we will see the borders of such cells. So border is here and here also. So this is one cell, cardiomyocyte. This is second cell, another cell, etc. This is the iron hematoxylin staining. As you can see in this diagram, the cardiomyocytes are branching cells. They have branches which are called lateral anastomoses. Lateral anastomosis, which allow the cardiac muscle cells to contact with each other and to form a network. And it is needed, this network is needed to provide the contraction of all the myocardium, of all the cardiac muscle cells together, because they should contract together to perform the function of the cell, of the heart, to pump the blood. So these anastomoses allow the cardiomyocytes to provide the impulse from one cell to another cell, which allow them all to contract simultaneously. This is the striated cardiac muscle tissue. It is striated because we can see here the cross striations, which are caused by a very high order of localization of A and I discs in myofibrils, as we have seen in muscle fibers in skeletal muscle tissue. The borders between the cardiomyocytes are called intercalated discs. These are the areas which have a few different cell junctions. The cell nucleus is usually one, maybe two cell nuclei for one cardiomyocyte. So the nucleus is located in the middle portion of the cell, just in the center of the cell. So here you can see the nucleus in the center of the cardiomyocyte. So these are the features of cardiac muscle tissue. We have here cells separated with intercalated discs. The cardiomyocytes are branching with lateral anastomoses. And feature number three, the location of nucleus. Nucleus is located in the center. Between the cells here we also have loose connective tissue with blood vessels and nerves. In this picture you can see the same. Also you can see here that cardiomyocytes are combined into longitudinal structures which are called functional fibers. And functional fibers are interconnected together with the help of lateral anastomoses. So this all will form the network, along which the impulse will spread from one cell to another cell with the help of the gap junctions in the intercalated disks. Cell nuclei are located in the center of the cells. We have intercalated disks and lateral anastomosis. In cross-section, we will see this picture. So these are cross-sected cardiomyocytes with nucleus in the center and cross-sected myofibrils, which look like dots. What is intercalated disc? Intercalated disk is a complex of intercellular junctions between cardiomyocytes. It is not flat, it has this specific form to increase the junction surface for better attachment of cardiomyocytes together. So this is the shape of its surface, and this is how it looks like in longitudinal section. So the junctions in intercalated discs include gap junction, zonular adherens, or belt desmosome, and desmosome. Gap junctions are these areas where two membranes of cardiomyocyte come very close together to form nexuses through which ions can pass from the cytoplasm of one cardiomyocyte to the cytoplasm of other one. So gap junctions are for communicating between the cells, and here they are also serve for transmission of electrical impulse, which is needed for contraction. Electrical impulse spreads along the membrane of cardiomyocyte, and it needs to go to the membrane of the adjacent cardiomyocyte, and this impulse can go through the gap junction. Zonular adherens, belt desmosome or intermediate junction, and desmosome are adhesive junctions for attachment, mechanical attachment of one cardiomyocyte to another one. Zonular adherens or belt desmosome or intermediate junction is located where myofibril of first cardiomyocyte should be attached to the myofibril of second cardiomyocyte. As you remember, zonular adherens junction include microfilaments in its structure. So here are microfilaments. These are actually actin microfilaments which make thin actin myofilaments inside myofibrils. So where you have thin actin myofilaments in myofibrils, there will have the zonular adherens, inside which we have actin binding proteins such as vinculin, taurine, and alpha-actinin in attachment plaque, and between the membranes of attaching cardiomyocytes, the integrate protein cadherins. So in this area, zonular adherens, in this zonular also zonular adherens, and here also the same, where we have myofibrils. Between myofibrils, in the area where we have some other organelles, such as smooth endoplasmic reticulum and mitochondria, in this area we will have desmosomes. It includes intermediate filaments in its structure, which go also along the cardiomyocytes between the myofibrils and serve for maintaining of the inner shape. This is how intercalated disc looks like in electron microscopy picture. So this is how it goes. It has some transverse component, which is vertical like this, and it has a lateral component, which is horizontal like this. In horizontal or lateral component of the intercalated discs, we usually have gap junctions. The same is here. This is the area where two membranes of the cardiomyocytes come close together. So here you can see membrane of one cell, membrane of second cell, and they come very close together to form nexuses or gap junction in this area. In the transverse component, we will have the zonal adherence junctions where we have myofilaments, myofilament here, and in the areas where no myofilaments are present, for example here, we will have the desmosome. The same is in this picture. This is the intercalated disc. In the area where we have myofibril from one cell and from second cell where they attach together, we will have zonular adherence also in this area and in this area. Here where you see the fusion of two membranes together, this will be the gap junction, and in the area where we have no myofibrils, for example in this area, we will have the desmosomes. Cardiomyocytes also have T-tubules and terminal cisternae of sarcoplasmic reticulum, but they form not diates but diates, not triates but diates. Diate means the structure which is composed of two elements, one T-tubule and one terminal cisterna. This is how it looks like in the picture, one T-tubule and adjacent from one side terminal cisterna of smooth endoplasmic reticulum. The principle of working is the same as for a triade in the skeletal muscle fiber. Also, we need calcium ions to get released from a sarcoplasmic reticulum to go to the troponin C molecule in actin myofilaments to launch the contraction. Calcium ions are stored in sarcoplasmic reticulum, smooth endoplasmic reticulum, which surrounds myofibrils, and in order to calcium ions to get released from smooth endoplasmic reticulum, we need electric impulse to go along the membrane of cardiomyocyte and along the membrane of T-tubule, and this signal transmits to the smooth endoplasmic reticulum, inside which we have the calcium channels that become open when the impulse reaches this area, and calcium can go out from smooth endoplasmic reticulum and reach the myofibril. So the mechanism is the same as for the skeletal muscle tissue. The difference between the skeletal and cardiac muscle tissue, also seen here in the structure of the myofibril in this picture. It has this 8, like number 8 shape, because the section went through the area of the branching of cardiomyocyte, like here. Here you can see that myofibril branches into two parts because of the lateral anastomosis present in this cell. And if we cut like this through this area, we will see that myofibril looks like this. And also the diates are located at the level of Z line. The triates in skeletal muscle tissue are located at the level between the lighter I band and darker A band. So triates would be located in this level. Diates are located in this level. We have 3 types of cardiomyocytes. The typical ones, most of the cardiomyocytes are typical or contractile cardiomyocytes or working cardiomyocytes. It is the same. So it is just ordinary cardiomyocyte, which major function is to contract. So it is branchy, so it has lateral anastomosis, intercalated discs, which combine contractile cardiomyocytes together, and most of its cytoplasm is filled with myofibrils. because it needs a lot of myofibrils to contract. Second type of cardiomyocyte is called secretory because of their function. Their function is to produce and to secrete the so-called natriuretic factor. And this substance is stored in secretory granules located in the central part of this cell. So the secretory function is its second function. The first function is also to contract. So secretory cardiomyocytes also can contract. This is why they also have a lot of myofibrils inside their cytoplasm. But they have additional function to produce natriuretic factor. And we have third type of cardiomyocyte, which is called atypical or non-typical, and we have subdivision of these atypical cardiomyocytes into the pacemaker cells or node cells. Their function is to produce electrical impulse, which is needed to heart for contraction. Second type of cells are cardiac conducting cells, and third type, the Purkinje cells, the cells of so-called Purkinje fibers. So pacemaker means the cell that make the pace, the amount of beats of heart per minute. It means that these cells, pacemaker cells, which are located in some areas in the heart, which are called nodes, these cells can generate themselves electrical impulses. So heart doesn't need nervous system to launch contraction, because cardiomyocytes themselves, these specific types, can generate electrical impulses. And they generate about 60 impulses per minute, and it makes our heart to beat about 60 times per minute. This is why they are called pacemaker cells. The cardiac conducting cells are smaller cells, and they conduct this impulse from pacemaker cells to the third type of cells, Purkinje cells, and they transmit this electrical impulse to typical cardiomyocyte, making them to contract. So here you see the typical cardiomyocytes. Here they are branchy with a lot of lateral anastomosis. You can see intercalated discs here, and one nucleus per cell located mostly in the center of the cardiomyocyte. Between cardiomyocytes, loose fibrous connective tissue. The same in oblique or cross section. This is the cross section or oblique section of one working cardiomyocyte. It is filled with a lot of myofibrils. Myofibrils are stained with eosin, and the nucleus is located in the middle portion of the cell. These are also the sections of cardiomyocytes. You cannot see the nucleus inside this part of the cell because this is because of the plane of sectioning. If you cut the cell like this, you will not see cell nucleus in its section. You will see cell nucleus only if you cut like this, in this area. So in our histological preparations, in cross sections, only in some sections the nuclei are visible. In others, nuclei are not visible because of the plane, because of how the section was made. The same in different staining, iron, hematoxylin. So these are longitudinal sections of cardiomyocytes, working typical cardiomyocytes with their lateral anastomosis, intercalated discs, and cell nuclei located in the central part of the cell. Another type of this histological preparation, the same. Iron, hematoxylin. These are the borders between the cells, intercalated discs. Here they look lighter. Cell nuclei not very well seen because they are hidden with the myofibrils. Myofibrils mask the cell nuclei. Here, for example, you can see the cell nucleus. You can see also cross striation. The same in cross-section. So here are the cross-sections of cardiomyocytes. In some of them, you can see cell nucleus in the middle part of the cell. Now, conducting cells. These cells provide the heart to have the so-called automatism, the ability to generate an action potential self-sufficiently and to contract. Action potential is the electrical impulse. It is the wave of the charge of the change of the membrane charge. So it is the change in the charge of the cell membrane which spreads along the cell. It is called the electrical impulse or action potential. It spreads along the cell and it can pass from one cell to another cell through gap junction. So the atypical cardiomyocytes provide automatism because they can generate the impulse themselves. In our histological preparation, we can see the cells conducting cardiomyocytes which form the so-called Purkinje fibers. Here we can see the structure of such a cell. It is round in shape, unlike the typical cardiomyocyte, which is elongated. It is much larger in size, and it has very few myofibrils, which are located chaotically, not organized, and mostly at the periphery of the cell. Here you can see these myofibrils located mostly at the periphery, and it gives such pushy appearance to these cells, such fluffy appearance, because of a few myofibrils at the periphery. In the central part of the cell, we have one or two cell nuclei and other organelles, such as Golgi complex, mitochondria, etc. This structure, which is made by a combination of several conducting cardiomyocytes, will be called Purkinje fiber. So, conduction system, which allows heart to contract, includes the nodes, sinoatrial node, atrioventricular node. These are nodes, the areas where we have pacemaker cells. They generate electrical impulse. They are located in these two areas. Next, these pacemaker cells transmit impulse to conducting cells, and next, conducting cells transmit impulse to the conducting cardiomyocytes, which form these elongated structures, which are called the His fibers, which are located here between the ventricles of the heart. The His bundle has two branches, the right one and the left one. So these are the His bundles, and then they branch into the Purkinje fibers that go along the wall of ventricles, spread between the endocardium and myocardium, and there the impulse is transmitted from conducting cardiomyocytes to working cardiomyocytes in the myocardium. So in our sections we see this area. It is where we can see the conducting cardiomyocytes of Purkinje fibers. So in our histological section we see a part of endocardium here, a part of myocardium, and between them goes elongated structure called Purkinje fiber. It consists of several large cells conducting cardiomyocytes. So here you can see a Purkinje fiber. It consists of conducting cardiomyocytes, and here you can see working cardiomyocytes. You can compare their size and shape. If you compare working cardiomyocyte with the atypical one, and also location of the myofibrils. They go along the working cardiomyocyte and fill all its inner space, but they are very rare and loosely packed, and mostly at the periphery in the conducting cardiomyocytes. This is how it looks like in our histological preparation. This is the section of the wall of the heart. You can see here the thin endocardium area. It consists of endothelium, the same tissue which lines the capillaries and other blood vessels from inside, and loose fibrous connective tissue. This thick area is the myocardium. It consists of contractile working cardiomyocytes. And here between endocardium and myocardium you can see this elongated structure, which is composed of multiple conducting cardiomyocytes, which are large and round cells. They are separated with the capsule made up with connective tissue. This is why it is so red in color. The same at high magnification. This is the endocardium, loose connective tissue, myocardium, inside which we see here working cardiomyocytes, and these are conducting cardiomyocytes. They are large, oval or round in shape, with one or two nuclei. In some cells, nuclei are not seen, they are not included in the section, and very thin pinkish myofibrils. The same is here, endocardium, conducting cardiomyocytes, myofibrils inside them at the periphery of the cells. This is how an atypical conducting cardiomyocyte looks like in electron microscopy picture. So you can see its nucleus, and myofibrils are not in high order of organization. Secretory cardiomyocyte, as the working one, contains a lot of myofibrils. Also it is branching. It has lateral anastomosis, it has intercalated discs, but its second function is to produce the atrial natriuretic factor. It is stored in secretory granules near the nucleus of the cell. So here you can see the part of the nucleus of the cell. These are myofibrils, lighter, stained with their Z lines, darker lines here. So this will be one the part of the myofibril. It's a structural and functional unit, a sarcomere. Between myofibrils you see darker mitochondria. You have here a lot of mitochondria connected together, and if you look at this area near the nucleus, you can see here a lot of dark secretory granules. Inside them we have this substance which is called atrial natriuretic factor. Atrial, because these cells which produce its substance are located in the atrium of the heart. What is the function of these cells? When our blood pressure becomes too high in the hypertension disease, for example, it is bad for heart to have too high blood pressure. So then the cells which are located in the atrium feel this increased blood pressure, and they start to release this substance, atrial natriuretic factor, in the case of increase of blood pressure. This substance goes to blood capillaries, and with the flow of blood it reaches the kidneys, and it acts upon the cells of the tubules of nephrons inside kidneys, making them to release more sodium and water with urine. So a lot of water is released from our body with urine. Urine is made from blood plasma, as you know. So if you release water with urine, it means that this water came from blood plasma. So the volume of blood plasma decreases. It means that the total volume of your blood decreases, and it leads to decrease of blood pressure if you have lower volume of blood in your blood vessels. So this is how this substance works. If you have too high blood pressure, atrial natriuretic factor is released, and it forces kidneys to release water from our body to decrease blood pressure. And now we go to our third type of muscle tissue. It is called smooth muscle tissue. Smooth because it has no striation. It looks like equally stained cytoplasm of the cells. The structural and functional unit is smooth muscle cell or smooth myocyte. It is elongated or spindle-like cell, elongated cell nucleus, one nucleus in the center of the cell, and smooth muscle cells usually form the bundles and layers. So here you can see how this tissue looks like in longitudinal section, where you see these elongated cells, spindle-like cells with elongated nuclei inside them. And in cross section you can see a lot of cross-sected red circles. Inside some of them you can see round cell nuclei. They are round in cross-section like this, but in some red circles you can see no nuclei because of how the section goes. If you cut like this, you will not see the cell nuclei in the sections here. If you cut like this, you will see here this cross-section of the nucleus. So here you can see nuclei, here no nucleus. So smooth muscle cells usually form bundles and layers. Inside the bundle, the smooth muscles are connected together with the help of intercellular junctions, and they are covered with the basement membrane, and between the cells we have loose connective tissue which is called endomysium, also with blood vessels, blood capillaries, and nerves. Here you can see the inner structure of a smooth muscle cell. So it has the cell membrane, basal membrane or basement membrane or basal lamina, the same. Together cell membrane and basement membrane form sarcolemma. Inside the cell, the cytoplasm is called sarcoplasma. One cell nucleus, it is elongated, and the feature of smooth muscle cells is that as all muscle tissues, it has contractile elements composed of actin and myosin myofilaments, but they are not very highly organized inside the cell. They do not form myofibrils. They are located, they go in different directions. You see them like this. Some of them go like this, some of them go like this, some of them go like this. These myofilaments are attached to the cell membrane with the help of the so-called cytoplasmic densities. They bind actin myofilaments of the thin myofilaments to the cytolemma. Inside the cytoplasm we have also the structures which are called the dense bodies. They also bind actin myofilaments and intermediate filaments to each other inside the cytoplasm. You can see here thin actin myofilaments, but myosin filaments in resting state are not present in the cell in the active state. They are non-active. They are depolymerized. Also, we need the calcium to contraction of smooth muscle cell, and the calcium is stored partly inside the smooth endoplasmic reticulum, in sarcoplasmic reticulum, and partly in specific invaginations of the cytolemma of the cell, which is called caveola. Caveoli are invaginations of the cytolemma of the smooth muscle cell for storage of calcium ions. You can see the same here, the structure of the smooth muscle cell. One nucleus in the center, some mitochondria, smooth endoplasmic reticulum, and caveoli are the places where calcium is stored. Here you can see myosin, red, and actin, yellow filaments needed for contraction. They go in different directions, not forming myofibrils, and they are attached to the cell membrane with the help of the dense densities, cytoplasmic densities, as you can see them here, and dense bodies inside the cell. So these bluish structures are intermediate filaments. They are also connected to the dense bodies with alpha-actinin, and also actin myofilaments are attached in the areas of the dense bodies inside the cytoplasm. The thick myofilaments, as in striated muscle tissues, are composed of myosin molecules with tail, neck, and two head regions, but unlike the skeletal muscle tissue, which has the area of the myosin myofilament which lacks heads of molecules, so in this region you see only tails, no heads. This is in striated muscle tissue, but this is in the smooth muscle tissue. You see that all the length of the thick myosin myofilament is covered with the heads of myosin molecule. This is the difference in the structure of the myosin myofilaments in striated and smooth muscle tissue. And the second difference is that in smooth muscle tissue, in resting state, the myosin myofilaments are not ready for contraction. So they are inactive. They look like this. When they are active, they should look like this. So in resting state, they are like this. They are dephosphorylated, and in order to for contraction, we need to activate them. So the mechanism of smooth muscle cell contraction. As for striated muscle tissue, we need calcium ions and ATP molecules for contraction. So calcium is stored in caveoli and in smooth endoplasmic reticulum. When some hormone or neurotransmitter binds to a receptor, it causes the calcium ions to go out from smooth endoplasmic reticulum and from caveoli into cytoplasma of the cell. This calcium next goes to a specific molecule which is called calmodulin molecule. So calcium attaches to calmodulin molecule. Then this calcium calmodulin complex attaches to the molecule which is called myosin light chain kinase. This molecule uses ATP molecule to phosphorylate myosin. So myosin goes from inactive to active form, And now myosin is ready to attach to the actin myofilament and to contract. Then happens the same as in the striated muscle tissue, so we will have the attachment of the head of the myosin to the actin myofilament, bending of the head of the myosin molecule, and movement of the actin uh, myofilament along the myosin one, and the contraction. In relaxed state, the cell looks like this. Inside it, you can see that myosin and actin myofilaments go in different directions. They are attached to the cell membrane at some angles, and when they are contracted, the cell starts to look like this. Very funny shape of the cell, and the cell nucleus, which is usually elongated, becomes spiral looking because of this funny shape of the cell. So you can see it, for example, in this picture. This is the electron microscopy picture of the cross-section of the arteriole. Arteriole is the small artery, the blood vessel. Inside blood vessel you can see erythrocyte and thrombocytes, platelets. These are endothelial cells. Endothelium lines all the blood vessels from inside. And the second layer here is composed of smooth muscle cells. And here you can see this funny spiral-like shape of the nucleus of this smooth muscle cell. So it's contracted form. The smooth muscle cells interact with each other because they are attached to each other to form bundles. Here is the cytoplasm of one cell, and the second cell is here. And here is the myocellular contact, the junction between two smooth muscle cells. This complex of junctions include desmosomes and nexuses. Desmosomes are for mechanical attachment of cells together, and nexuses, gap junctions, are to transmit impulse and small ions from the cytoplasm of one cell to the second one, to allow them to contract together. Innervation of smooth muscle tissue is shown here. The smooth muscle tissue is innervated with the autonomic nervous system, unlike skeletal muscle tissue, which is innervated with somatic nervous system, which allows voluntary contraction. Smooth muscle tissue is innervated with autonomic nervous tissue, autonomic nervous system, which allows only involuntary contraction. As you can see here, some smooth muscle cells are innervated with, when each cell is innervated with its end of the axon of the neuron, and it is called multiple innervated smooth muscle. So each cell gets its individual innervation. It is for fascic muscles to have relatively high speed of contractions. But most of the smooth muscle tissues are innervated with single terminal ends of axons of the autonomic neurons, and in these areas of the end of the axon, the neurotransmitters are stored, and when they go out from these areas, they act upon their receptors upon the membrane of this cell. This cell becomes excited, and it spreads the impulse through gap junctions to other cells in this bundle, and as the result, they will contract all together because of gap junctions between the cells. Where we can see smooth muscle tissue, we can see it in inside the wall of blood vessels and inside the walls of some organs. We see the smooth muscle tissue in the sections of small intestine or in the section of the stomach. As you can see here, in intestines we have two layers of smooth muscle tissue, which allows the intestine to contract while peristalsis happens, and it allows food to be moved along the way of the intestine. So in the outer layer the smooth muscles are oriented longitudinally. In the inner layer smooth muscle cells are oriented concentrically, like this. It means that if we make the cross section like this, in the inner layer we will see longitudinal sections of smooth muscle cells, and in our outer layer we will see the cross sections of smooth muscle cells. This is how it looks like in our histological preparation. So this is the inner circular layer of smooth muscle cells. Here you can see elongated smooth muscle cells, longitudinal sections, and this is outer layer of muscle. Here you can see cross sections of them. The same is here. In outer layer cross sections, in inner layer longitudinal sections. The same at high magnification. You can see here elongated spindle-shaped cells, and here cross sections of them. Another preparation of the same, also elongated cells with elongated nuclei, and here they are in cross sections. Some of them have sected nuclei, some of them do not have nuclei in this particular section. Also we can see smooth muscle tissue in the walls of blood vessels, for example in arteries, which have a lot of smooth muscles in their walls. So this is the section of the artery with erythrocytes inside. This is endothelium, and this is smooth muscle tissue. And here you can see this specific funny spiral shape of cell nuclei in smooth muscle cells. So these are the cell nuclei of smooth muscle cells. So it was all for today. Thank you for your attention. See you in class.
Спасибо вам.